Jai Jinendra. Hello friends, welcome to Wings to Dreams. So today I am Khushi Jain. According to our ophthalmology section, continue with the today's topic, that is disease of cornea. So in this, let's first of all study with the disease of cornea, and that is first part is anatomy and physiology of cornea. So friends, if you like my channel, please subscribe, share, and don't forget to tap the bell icon also. So let's start with the current today's topic, that is cornea. So first of all, what is eyeball actually? It's what is cornea? It's a outermost covering of the eyeball, or we can say epithelium of the cornea. Actually what we have is our eyeball is consist of three layers: outer, middle and inner layer. So outermost layer has a part, first part is known as cornea. Cornea is basically a avascular part, okay? It's avascular thickening which is also transparent, which we can't see. So first of all, our outermost layer of the eyeball consists of three parts: the corneal, the sclera and the limbus. Here the sclera is the whitish part you can see in the eyes. The limbus is known as the junction between the where the cornea and the sclera actually meets. Okay, so that junction is not particular its point. Everywhere in the eye, the junction of the cornea and the sclera is known as limbus. And the cornea is basically what the transparent film present uppermost layer of the eyeball is known as cornea. Okay, so let's start with the studying the basic anatomy and physiology. After it, I will continue to another kinds of disease of cornea. So cornea is avascular, as I told you, and it's a transparent and glass-like. Avascular here is what mean avascular, that is less of blood, that there is no blood supply. Okay, then transparent, and it's a glass-like structure. After the dimensions are what the cornea is anteriorly elliptical, posterior surface is adhere to the posterior parts. After that, what there are other dimensions also: anterior, posterior, inferior, superior, and refractive index of the cornea is 45 millimeter approx, or I can say 45 diopter. Yeah. So after that, nerve supply. Nerve supply of the cornea is by the nasociliary nerve. As you all know, if you know the anatomy, nasociliary nerve is the branch of trigeminal nerve, ophthalmic trigeminal nerve, branch of trigeminal nerve, fifth division, in which ophthalmic branch is of the nasociliary nerve. Okay. After that, most important thing what we have to study in this whole lecture, or I want to focus, is the basic this thing, the histology of the corneal epithelium. Okay, so histology, it cornea, what you see, like it's a just transparent film. I told you before, na. So it's a just transparent film, and it have 6 layers. ओके, सो ये हम कह सकते हैं कि ये कोई ट्रांसपेरेंट फिल्म है, एक पतली सी लेयर है, लेकिन इसके बावजूद भी इसके पास ऐसे 6 लेयर्स है जिसकी वजह से हमारी आई को प्रोटेक्ट कर रही है. So what are these 6 layers? These are epithelium, Bowman's membrane. After that, there is a corneal stromal layer, pre-descent membrane, descent membrane, and the endothelium. So we will deal in our video with all these epithelium one by one. And especially I want to focus you that you have to learn this much. Most important thing is this epithelium and its membrane, so that we can learn what are the disease, what are the anomalies. If you know what are the functions of these 6 layers, what are the functions also, which kind of layer can regenerate, which cannot, what is the functioning, what are the features of each of the layer. After you know this much, you can easily be deal with and understanding the level of disease of cornea. And this part is as much as important for your theory section as well as your for practical examination and for further PG level. Okay, so let's start with the next, that is the layers of the cornea. ओके, सो कॉर्निया की लेयर्स के बारे में अगर हम डिस्कस करें, फर्स्ट ऑफ ऑल लेयर इज द एपिथेलियम. Epithelium is the outermost layer of the cornea actually. Cornea is the outermost layer of the eyeball, okay, and epithelium is the outermost layer of cornea. Got it? So now in case of epithelium, there are 3 types of layer in the epithelium membrane also. That is the superficial, middle and basal layer. I have made it from diagrammatic representation. Here I want to tell you that the basal representation, that is of the columnar epithelium cells, Uppermost superficial and middle layers are quite like flattened type of the cells, and the lowermost, that is the basal layer of the epithelium, is the columnar type of the cell. Okay, so that is the most important thing. That is why the corneal epithelium has a power of regeneration. Regeneration is basically what, if any kind of, due to any kind of destruction, any kind of, I can say, infection or any kind of etiology, idiopathical causes, if the layer of the epithelium get destroyed, then the basal layer has cells have ability to mitosis, ability to perform mitosis, and if they perform the mitosis, then they can regenerate their epithelium cells. And what is this? This is the epithelium, non-categorized, stratified, squamous epithelium. That is, I use here short form NKSS. So epithelium is basically formed of non-categorized, stratified, squamous epithelium, and it is the power of regeneration. That is the basal. And of course, it's the outermost layer. So what would be its function? It would prevent or any kind of foreign body particles. Okay, so normal epithelium जो हम basically अगर हम देखते हैं कि normal कचरा चला जाता है तो आंखों में जलन होने लगती है. So outermost for protection, foremost protection is given by this epithelium only, that is a epithelium non-granulous stratified squamous epithelium. After that, the second layer of cornea is the Bowman's membrane. Yes, Bowman's membrane is what the specialty is. It's a acellular type of membrane. Second layer of the cornea is acellular. It has not the ability to regenerate. Okay, if it get destroyed, it can't regenerate, and it is a periodic acid shift stain negative. What here I mean, that it cannot stay. You have here and staining types, various types of staining. So if we stain this layer in the microscopy, then it would be result in negative. So I have written this PAS negative, that is periodic acid shift stain is negative. And scar. Scar is what, if any kind of injury occurs to the Bowman's epithelium, then it will heal with the help of scar. And of course, if there is a scar, then it will lead to opacity also. Opacity is basically, you can say कि हमें आर पार कुछ नहीं दिख रहा. Like हम कह रहे हैं कि कॉर्निया इज ट्रांसपेरेंट, लेकिन अगर फिर भी हमें आर पार अगर कुछ ना दिखे, लाइक अगर ऑपेसिटी हो जाए, तो उसे हम बोलेंगे स्कारिंग के हील होने के बाद जो रहती है. ठीक है, सो ऑपेसिटी इज ऑफ थ्री टाइप्स, नेबुलर, मैक्युलर एंड leukomatous opacity. Other, if we talk about the nebular opacity, it is a mild faint opacity. Macular and the macular is mid, mid type, and the leukomatous is the furthermost. We can say full opaque zone is seen. Okay, so person is not able to see actually what is not watch actually what is surrounding, what is in its surrounding area. So there is a leukomatous opacity. Now here a question comes that which type of opacity is mostly impair our vision. Okay, so I told you that there is opacity, full opacity in case of leukomatous. So you all will be answering the leukomatous, yeah. But no, I have asked which kind of opacity may be result in impaired distortion. That is, distortion is vision is intact. Okay, so nebular opacity, the first one, will lead to vision intact. That is most of the vision, because in leukomatous there is all opaque everything, but in case of faint opacity, the person also feel pain and something else, irritation, discomfort. So mostly then nebular opacity will be presenting the discomfort. And if the question turns, and it will become like which kind of opacity you will have the maximum frontalness, or you can say maximum darkness or halos around the eyes, so then you can give the answer to the leukomatous. Okay, so guys, remember for this point, scar leads to opacity. I have not included as much what I am saying in my board, because I hope that you all are understanding my points and you are writing or not downing my points. That's why. So after that, we will deal with the third layer, that is the stromal layer. Stromal layer is the most important, we can say. It is the third layer, and this is the strongest layer of it contains, it is the widest layer, I can say, not strongest, it's the widest layer, I can say. It contains 90% of the part of the epithelium it contains 90% part of the whole of the cornea, corneal layer. Okay, it contains what? It contains collagen fibers. There are various types of collagen: collagen 1, 2, 3, 4, and 5 to up to more. But most common type of the collagen fiber which it contains is a type 1 collagen fiber. Okay, after that there are glycosaminoglycans. Basically, you know the proteins. So these are commonly, which are also commonly present on ananas, chondroitin sulfate, keratin sulfate, and much more. But these two are the most common. That is chondroitin sulfate and the keratin sulfate. So this is all about the stromal layer fibroblasts I have written here. So what's that mean? I want to show here. That is the it has performed the prevention. It has performed the function of maintaining stability to the layer of the cornea, all layers of cornea, and also it is a fibroblast cells that is helps in the phagocytosis or any kind of foreign particles, and it can leads to help in prevention of infection. Okay, with the help of these type of cells. After that, what we have the Duas layer. Duas layer is the fourth layer, as you can see. Duas layer, why its names are Dua? Dua. So there was Dr. Harvinder Dua. So in behalf of name of it, because he discovered this layer. So from this Dua, there is a name of this layer known as Dua's layer. Okay, it is the strongest layer. Dua's layer is the strongest layer. If you can say अगर आपके कॉर्निया में कोई इन्फेक्शन हुआ है, कोई एब्नॉर्मलिटी आई है, तो आपकी जो लेयर्स है वो इन्फेक्ट होना शुरू हुई. ओके, कॉर्निया जो इन्फेक्शन होना शुरू हुआ, एपिथेलियम पे गया, बाउमेंस पे गया, स्ट्रोमा पे गया. लेकिन अगर उसने डुआज लेयर को भी, और फर्दर वी विल स्टडीइंग द प्रीडेस्मेंट मेम्ब्रेन एंड डेस्मेंट मेम्ब्रेन. सो डुआज लेयर को हम प्रीडेस्मेंट मेम्ब्रेन भी बोलते हैं. तो अगर उसने इस लेयर को डिफेक्ट किया, इफ दैट पार्ट इन्फेक्शन इज कॉजिंग दिस एंड this strongest layer, because it is the strongest layer. How much? It is a 15 micrometer thick, or I can say 14, 70 to 17 to 7 to 14 micrometer thick. So if the infection will reaches to this layer, then हम ये कह सकते हैं कि हमें अब केरेटोप्लास्टी, और हम कहें कि हमें अब सर्जरी की नीड होगी ही होगी. ओके, सो अगर इतना है तो हम कह रहे हैं कि रीजनरेशन हो सकता है एपिथेलियम लेयर का, लेकिन डुआस लेयर के अंदर वी हैव टू डू द सर्जिकल. सो हम यही ट्राई करते हैं एज अ क्लिनिक एंड ओपीडी दैट डुआस लेयर वुड नॉट अफेक्ट द दिस पार्ट, कॉर्नियल पार्ट. Okay, so this is all about the four layer: epithelium, Bowman's membrane, stromal layer, and the Duas layer. It's a 15 to 15 micrometer thick. Okay, after that, I hope this will be clear to all of you about the four layer. After that, I will come to let's start with this layers, that is descent membrane and the endothelium. Okay, so descent membrane is basically the strong layer, and it is a power to regenerate also. Another word, which layers are regenerating? Yeah, there is epithelium layer, in which basal part, because of the basal layer which have ability to mitosis, was regenerating. Also the Bowman's membrane do not regenerate. After that, this descent membrane can regenerate. It has a peripheral termination of descent membrane known as Schwab's line. Okay, so basically it is usually related with the glaucoma. Okay, if there is a peripheral termination of this descent membrane in our eyes, it will make a line like structure. We say it as a Schwab's line. Here you remember just it as it is. If we study, you have studied the glaucoma in pharmacology, or you are studying further in ophthalmology, we will deal with this topic, Schwab's line, in whole in the glaucoma topic. Okay, so desmetocin is basically what? If the descent membrane, usually it's not found, but in case of infection, in case of bacterial corneal ulcer, desmetocin formation occurs. What is it actually? When we will study bacterial corneal ulcer, we will deal with this also, but here I want to give you just a brief of it. Is what? If any kind of aqueous humor or any kind of fluid occurs in the anterior chamber, or protrudes to the infection occurs to the descent membrane, now that it will lead to the formation of desmetocele. Desmetocele is basically cele means cavity, so it's a formation of cavity in the descent membrane known as desmetocele. Okay, so this is all about what also it's a strong layer, but strongest was Duas layer. Okay, descent membrane. After that, we will deal with the endothelium. Endothelium layer is a single cell layer, and it is the innermost and the posteriormost, we can say, layer of the corneal epithelium. It is metabolically active. What is the function? I told you we have to learn what are the membranes, layers of the corneal epithelium, and what are the functions of each. Okay, so in case of this corneal epithelium, endothelium endothelium. It's a metabolically active layer. And what is metabolically active? That is, its function is metabolic metabolization. Okay, we can say here. So what is this? This is a transparency maintained. It is the main important part of the cornea which maintains the transparency of cornea by two mechanisms. That is, first one is by active sodium and potassium pump mechanism, and second one is tight junction barrier. Okay, so sodium and potassium pump mechanism is what? That endothelium layer, which is the innermost layer, it is a pump mechanism of sodium and potassium. Also, there is sodium bicarbonate, hydrogen, potassium. Also, I have written this only once. They are as much as pump. They maintain the cellularity. We can say they maintain the mechanism of transparency of the cornea. Also, what there is a tight junction between both the adjacent cells. Tight junction, when there is two cells adjacent to each other, are tightly binded to each other. We can say there is a tight junction. So this tight junction will prevent the any kind of light, or we can say any kind of object to go into the innermost layer or the outermost layer. That is, it maintains transparency. Okay, it maintains its whitish part or glassy halo appearance. It acts by acting as a barrier by making the junction, that is tight junction between them. After that, endothelial cells, it has a normal cell count. How much? It's about approx to 2400 to 3000 cells per meter per millimeter square. Okay, now if this cell count is increased or decreased, there would be also deformity, or we can say there is also any kind of infection, or it would indicate any kind of abnormality. So if it occurs that if this cell count will increase to more than 3000 cells per millimeter square, it leads to polymorphism, polymagnetism. Okay, it will enlarges the cells, and if it decreases, that it will decrease less than 2400, then it will lead to the edema of the epithelium layer. Okay, after that, what the thing is the investigation. After this whole part, the whole endothelium would be investigated by which instrument? It is the specular microscope, which we commonly use. Okay, after that, transparency. So before this, I want to say you, this is all about what the basic anatomy of cornea, dimensions of the cornea, what are the layers of the cornea, and what is the function of the cornea. I hope you all remember it. And this content is basically from the ophthalmology textbook only. Okay, so after the transparency is basically, cornea is avascular, as you all know, and I told you first, it is transparent also. So what are the factors that includes its transparency? What are the factors that are maintaining its transparency? Are as follow. Further two factors I have written already mentioned here, that is active sodium potassium pump and the tight junctions. Okay, so what are the another factors? Another factors are tearful, avascularity of cornea, IOP is the intraocular pressure. After that, what is a evaporation, crystalline protein, water soluble protein, corneal lamella. Okay, so let's come to this one by one. First of all is a tear film. Tear film is what basically a film formed by lacrimal gland. We will deal with this also when we study in detail about the lacrimal apertures. Okay, so tear film is basically also helps in maintaining the corneal transparency. Avascularity of cornea, as you all know, it's a there will be loss of blood supply, so it's also maintain the transparency. After that, intraocular pressure. Yeah, if no, there is normal intraocular pressure. Intraocular pressure is what the pressure between the aqueous chamber, anterior and posterior chamber, anterior segment and posterior segment of what aqueous humor and the vitreous humor is that you all know now. Okay, so intraocular pressure जो है वो अगर maintain रहता है, normal maintain रहता है, तो वो क्या करता है, transparency को भी maintain रखता है. After that, evaporation with the outermost layer of the cornea also helps in preventing the evaporation, so that dryness will not occur. It will be remain as transparent. Okay, so this feature also helps with crystalline proteins. Crystalline protein is what basically are the water soluble proteins, जैसे कि keratin sulfate or keratin precipitate, we can say keratocytes, we can say they are also helping maintaining the transparency of cornea. And corneal lamella, that is laminal structures of the peripheral cornea, or all these layers helps in maintaining a transparency of cornea. So guys, this is all about my today's topic, that is anatomy and its basic physiology of cornea. If you have any kind of doubt and any kind of detail you want to or anything you want to ask, please comment on you. Please ask me on your comment section. And if you like my video, then please subscribe, share, and tap the bell icon also. This is not just for me and you. This will increase my courage also. It will increase and it will helpful, and it will motivate me more to increase and make much more videos, so that I can help you at free of cost. So I have motive. My motive is basically to give you and spread knowledge. So just please like, subscribe, and share my channel, and as much as you can, it would be helpful for you. I will be thankful for that. Okay, so thank you so much.